to our Science Squad channel. And for a few days ago, some sort of outbreak has happened among the staff at NMU. Faculty is breaking out in random dance during the class and singing at inappropriate times, and they cannot stop themselves. For help from CDC, we have determined that there is a bad case of RAD disease goes around campus, and there is one catch. Students are immune. Since the CDC is full of professional adults, we were giving an assignment to figure out what exactly the RAD disease is and how we can stop it. Now let's welcome 3 students from MSED 251 are on the team to treat the RAD disease.
My name is Myra.
I'm Mady.
I'm Anna.
Let's go, Myra, and hear about the source of the pathogen.
To get to the bottom of what exactly was causing the RAD disease, we first had to find the source of the outbreak. With our prior knowledge of only staff in the SELPS department being those infected, our first step to uncovering the source was complete. Using this information, we used an epidemiology chart to try to reason out our sources. Some questions we asked were, was it a single person who started it? Did it come from pets? What were the job titles of those affected? Some commonalities among those infected were that they were coffee drinkers and worked in the office or as teachers. Using this data and the process of elimination, we were able to come to a conclusion. The pathogen was spread by contact with the office coffee pot. Although not all victims drank coffee, they all had contact with the pot in some way.
Now we know the source of the outbreak, and we need to find out exactly what type of the pathogen it is and how it affects the body. Here is how we did it.
We discovered that it was a non-living virus based on the fact that it was only spread by touch and cannot reproduce on its own. It can only come from the direct source, while other pathogens can spread via body fluids or being in the same environment as the infected. RAD does not seem to spread so easily. We traced it back to the coffee pot in the teacher's lounge that each of the infected had come in contact with. Past direct contact with the coffee pot, the disease has not spread due to the type of pathogen and also the quarantine. In the beginning, it was important to know where RAD was active so that we could get a better idea of what we were dealing with so that researchers could start a treatment plan. We did this by taking cell samples from both the healthy and the people affected by RAD. We took samples from each system and tested them against each other. We saw that it affected the nervous system, which made sense due to the fact that it was causing involuntary movement all over the body.
Knowing which organ system is affected is very important to begin to narrow down a treatment plan. Since the treatment goes down to a microscope level, we must next look at the tissues and cells and how they have been damaged by RAD disease.
As we now know about the organ systems and how they function, we can dig a bit deeper into how exactly the RAD disease is affecting the body. We understand that the virus mainly affects the nervous system and subsequently the musculoskeletal. The tissue that the virus is affecting is nervous tissue. When nervous tissue is damaged, it cannot renew or revive itself. Nerve cells are responsible for sending electrical and chemical impulses throughout the body to help tell each system what to do. In our case, the damaged nervous tissue is causing random acts of dancing, singing, and head scratching, which is uncontrollable. Because it is uncontrollable, we know that the nervous system is not doing its job. This also affects muscle tissue as it is what helps the patients dance.
It is important for us to understand how a healthy cell functions before we can able to understand what might be wrong with cells when it comes to the RAD disease. Now we will hear from Anna as she showed us what the model of a healthy cell looks like.
All right, here we have the system model of what a healthy animal cell looks like. So the various main components within the cell are referred to as organelles. These organelles and their functions are all closely related. Many of the organelles rely on each other to accomplish their respective tasks. So, when one component of the system fails, many other components fail. In short, all the components of this system are interconnected in some way. Now that we have a solid understanding of the relationships between the cell components, I'll quickly go over what exactly some of the functions of the organelles are. So here we have the nucleus. This contains the cell's DNA and also just kind of organizes what the actions of the cells are. Next, we have the mitochondria, known commonly as the powerhouse of the cell. It also takes in and breaks down nutrients in order to create energy for the cell. Here we have the Golgi body. The main function of this organelle is processing and sorting proteins. Here we have the endoplasmic reticulum. which helps, like the Golgi body, helps produce and transport proteins. Now we have the lysosomes, which help remove waste from the cell. And then surrounding this whole set of organelles, or the whole set of components, we have the cell membrane. The cell membrane actually serves as the boundary in this cell model. The cell membrane is semi-permeable, which means some things can pass through the cell while others cannot. The membrane's permeability influences what inputs are able to enter the system. For example, common inputs include nutrients or proteins, which are actually able to pass through the selectively permeable membrane. On the other hand, outputs include waste or bacteria, which are able to be flushed out the inside of the membrane from the inside of the cell. So those are the outputs and inputs of this system. Though it is microscopically small, the cell is complex and definitely plays a crucial role in our eventual discovery of what causes the RAD disease.
Now that we understand how a cell should function, what does a model of an infected cell looks like?
Nerve cells are long and connect to each other via the myelin sheath and the axon, which transfers information between two cells. People affected with the RAD have a gap in their myelin sheath, which compromises the information and it gets lost. The disease attacks by coming in through the cell membrane over here. Once it is in, it travels to the nucleus and tricks the nucleus into copying the disease's DNA instead of the cells. With this, more pathogens are created, and once there's too many, the cell will burst and spread the diseases to other cells, and the process continues from there.
It is clear that the RAD disease is complex. What are the tissue engineering designs that have been selected to try to treat the RAD disease?
All right, so we've chosen to use stem cell injection and the nerve tunnel tissue engineering designs to direct our treatment of the RAD disease. So our first method that we chose is the stem cell injection. Stem cell injection involves injecting stem cells into the damaged area, which in the case of the RAD disease is the brain. Eventually, the stem cells will develop into brain cells in order to replace the cells damaged from RAD. Of the tissue engineering procedures available right now, stem cell injections seem the most practical. Most of our information about stem cell injections have been provided by scientists at University of Pittsburgh and Harvard. These researchers have completed several successful clinical trials of stem cell research, which shows this treatment plan may have success with treating RAD as well. Additionally, the fairly non-invasive nature of the stem cell injection sets it apart from many of the other treatment options available. The second treatment plan we're relying on to treat RAD is known as the nerve tunnel tissue engineering. RAD has damaged nerve cells, which is preventing the nerve cells from sending signals and messages to each other. The nerve tunnel involves implanting a plastic tunnel to direct new tissue growth in the damaged area. The plastic tunnel will help direct new nerve tissue growth in the correct direction, thus making it possible for the nervous system to send signals.
So, how are scientists use both stem cell injection and the nerve tunnel to create an optimized design? How this optimized design help treat the RAD disease?
Our optimized treatment plan involves potentially relying on both stem cell injections and the nerve tunnel. First, patients suffering from RAD will undergo the stem cell injection. As mentioned before, through this procedure, stem cells are injected into the brain, which is the area damaged from RAD. The stem cells being injected are made from the same patient's skin cells. According to Harvard and Pittsburgh University scientists, stem cells should eventually develop into nerve cells. However, research has also shown the stem cell injection process is not perfect. In some cases, most of the cells have died after injection or misaligned with the damaged tissue, which has prevented the success of the treatment. To further understand this treatment, we can understand that the boundary is the membrane, the injection is the input, and the relationship is how the stem cells and the damaged nerve cells react. If the stem cell injection proves to be unsuccessful, we will then turn to the nerve tunnel, which is a more invasive process. If the stem cell injection does work, the patients will only have to undergo injections rather than
So since the nerve tunnel implantation is slightly more invasive than just a simple injection, we are using it kind of as a plan B and second phase of our treatment plan if the stem cell injection does not work. So looking here, we have the model of the nerve tunnel implantation. essentially, a plastic tube is put between the gap in the nerve cells. Putting this tube in allows signals to be sent between the different nerve cells, which is unable to happen prior to this implantation due to the deterioration of the nerve cells due to RAD. So once this plastic tube is put in, the nerve cells are able to regenerate and grow in the correct direction from this cell to this cell in order for signals to be sent to and from. This does require surgery to be implanted. And in this model, just looking at just to understand it a little bit more, we can see the boundaries. The general boundaries of this system are the membranes of the various nerve cells. The input in this model is, again, the plastic tube put in to help facilitate the regrowth of the nerve cells. And then the general relationship between the components, which the components are the different nerve cells. The relationship is that all the nerve cells ideally are supposed to intersect with each other, connect, and a signal should be sent all the way across all the nerve cells in order to properly send signals to the rest of the body.
Okay, so thanks for 3 students from MSED to 51 students, and now we know the source of the pathogen, how it affects our body, and the optimized treatment plan. If you want to know more information, you can visit our Science Squad channel on YouTube. Thanks for watching.